Now, in this part of the discussion regarding neuronal transmission, we shall discuss about the mechanism of chemical transmission that takes place from one neuron to another. Now, from our previous discussion, we know that neuronal transmission is a process which is a combination of electrical transmission as well as chemical transmission. Till now, we have discussed in detail about the electrical transmission and the generation of action potential or nerve impulse, which travels along an axon of a particular neuron until it reaches the synaptic terminus. We have also discussed the various factors which determine the nature and the frequency of the action potential. Now, in this presentation, we shall discuss about the chemical neurotransmission, which is the transmission across the gap or the synapse between the presynaptic cell and the postsynaptic cell. Now, how many kinds of neurotransmitters are there? There are more than 50 kinds of neurotransmitters which has been identified, and each of these neurotransmitters have their specific receptors. So as we have discussed before, the neurotransmitters are packaged inside vesicles at the presynaptic terminal. So when a nerve impulse reaches the presynaptic terminal, these neurotransmitter filled vesicles fuse to the presynaptic membrane, releasing the contents, that is, these chemical messengers, to the synaptic cleft. Following these neurotransmitters bind to their appropriate receptor and influence the activity of the postsynaptic neuron. Now, the next question is, does a particular neuron produce multiple kinds of neurotransmitters? Yes, some neurons are capable of producing or releasing more than one kind of neurotransmitter at different times, depending upon their stimulation frequency. Then other question is a neuron capable of simultaneously releasing two kinds of neurotransmitters? The answer is yes. In certain cases, two neurotransmitters are packaged within the same vesicle. So the fusion of that vesicle leads to the simultaneous release of two neurotransmitters into the synaptic cleft. Now, this capability of a single neuron to release more than one kind of neurotransmitters makes the neuron capable of bringing about different influences on the postsynaptic neuron. Now, coming to the types of synapses, now there can be two types of synapses. One is the electrical synapse and another is the chemical synapse. In the electrical synapse, there are direct channels or gap junctions which connect the cytosol of the postsynaptic and the presynaptic neuron. So when the action potential reaches the presynaptic neuron, there is a direct exchange of ions through these gap junctions, and therefore the neuronal transmission is, in this case, a purely electrical event. The neuronal transmission across the electrical synapse is extremely fast, and such synapses are present in the hippocampus, which is a region of our brain which is responsible for our memory, and is also observed in the embryonic nervous tissue. Now, apart from the electrical synapse, there is also the chemical synapse. The chemical synapse is our more familiar kind of synapse, in which the neurotransmitters, in response to the nerve impulse, is released via exocytosis into the synaptic cleft, and after this is the most common type of synapse, and after the release of the neurotransmitter, it can either bind to a channel, in the case of channel linked receptor, or bind to a G protein coupled receptor, which we will be discussing shortly. Now, these chemical synapses, the transmission across these chemical synapses is however slower than that of the electrical synapses, because time is necessary for the neurotransmitter to get released, for the diffusion of these neurotransmitters in this synaptic cleft and their appropriate binding to their receptors, and therefore a synaptic delay is observed in the case of chemical synapses, which is slower than the transmission across electrical synapses. Now, speaking about the receptors which are present on the postsynaptic neuron. So the neurotransmitters are capable of binding, depending upon their specific nature, they are capable of binding to channel-linked receptors or G protein coupled receptors. Now, when a neurotransmitter binds to a closed channel-linked receptor, upon ligand binding, the channel opens, allowing the passage of ions across the membrane, and this leads to a change in the membrane potential. The ion channels are open instantly and they remain open for 1 millisecond or less until till the ligand is bound. Now, these channel-linked receptors can either be excitatory or inhibitory. Now, these are the different kinds of names of neurotransmitters like acetylcholine, glutamate, aspartate, ATP. These are excitatory neurotransmitters. So these neurotransmitters, when they bind to their appropriate receptor, these these receptors are usually cationic channels. For example, when they bind to the receptor, if they allow Na plus ion entry into the cell from the extracellular side to the cytosolic side, then this will lead to the depolarization of the membrane potential and therefore lead to an excitatory postsynaptic potential. In contrast, there are neurotransmitters which bind to the inhibitory channel linked receptor, for example, the neurotransmitter GABA or glycine, they bind to the corresponding receptor, which is actually an ion channel which allows chloride ion to pass from the outside to inside. This leads to the further reduction in membrane potential, hyperpolarization, and hence inhibitory postsynaptic action potential is induced in the postsynaptic cell. Now, coming to the next kind of receptor, which we said is the G protein coupled receptor. These receptors are actually transmembrane protein complexes, which is shown here, which are coupled to the G protein on the cytosolic side. The activation of these G protein coupled receptor upon binding to the neurotransmitter leads to a wide range of metabolic changes inside the postsynaptic cell, and hence these receptors are called metabotropic receptors. Now, what are the specific kinds of changes which happens in the postsynaptic cell? Now, once the neurotransmitter binds to the receptor gets activated. This activates the associated G protein, which is present on the cytosolic side of the receptor. This G protein now binds GTP and then goes and activates an enzyme adenylate cyclase. What adenylate cyclase does is it acts on ATP and converts it to cyclic AMP. This cyclic AMP is a second messenger, and this cyclic AMP can go back to the membrane, and it can change membrane permeability by opening and closing of ion channels. This same cyclic AMP can also cause activation of certain enzymes. The cyclic AMP can also go inside the nucleus and activate expression of certain genes. Now, therefore, depending upon whether the neurotransmitter is inhibitory or excitatory, depending upon the chemical nature of the neurotransmitter and the receptor on which it binds, whether it is a G protein coupled receptor or a channel linked receptor, diverse kinds of activities can be initiated in the postsynaptic cell. Now, we can also based on our discussion till now, classify the neurotransmitter based on its function, that is, whether it is excitatory or inhibitory, or based on its speed of action. That is, we mentioned that the neurotransmission across electrical synapse is faster than that in which the G protein coupled receptors are involved. Now, in this diagram shows three different types of G protein neurotransmitters, which all of them bind to the G protein coupled receptor and stimulate different kinds of G proteins, and thereby they initiate different kinds of pathways or chemical reactions inside the cytosol of the postsynaptic neuron. Therefore, these have very different consequences depending upon their different targets of action. The binding of these neurotransmitters, all these neurotransmitters, although they bind to G protein coupled receptor, can have very different influences on the postsynaptic neuron. Now, can these neurotransmitters bring about long lasting changes in the postsynaptic neuron? Yes. This is done by influencing the expression of the genes in the postsynaptic neuron. Now, how does this take place? When the neurotransmitter binds to the G protein coupled receptor, it leads to the activation of the G protein, the activation of adenylate cyclase, and the production of cyclic AMP. The cyclic AMP goes and binds to protein kinase A, which is a phosphorylating protein, which phosphorylates other proteins, leading to its activation. Now, presence of electrical signal can also open a calcium channel, which leads to the inflow of the calcium ions, or a presence of a hormone can activate a receptor tyrosine kinase, a particular kind of receptor, and both of this leads to the activation of a particular kinase enzyme, which is the MAP kinase. Now, both these activated protein kinase A and MAP kinase can go and phosphorylate transcription factors which are sitting upstream of specific genes. Now, this phosphorylation of these transcription factors switches on the expression of these genes, leading to their transcription and translation, and therefore these specific proteins now become available inside the cell. These can lead to long-lasting changes in the postsynaptic neuron. Now, looking into in a little bit more detail on the mode of action of a inhibitory neurotransmitter like gamma aminobutyric acid, what happens in this case is gamma aminobutyric acid or GABA can bind to the GABA A receptor, which leads to the opening of the chloride channels, and the chloride channels rush in. The inside of the cell becomes more negative. The cell membrane therefore the inside becomes more negative and is more hyperpolarized, thereby giving rise to a IPSP. The GABA can also bind to the GABA B receptor, and this can cause the inhibition of the voltage gated calcium channel. The low availability of calcium can interfere with the fusion of the synaptic vesicle to the presynaptic membrane. The binding of GABA to the GABA B receptor can also lead to the opening of the potassium channels. The K plus ion move from the inside the cell to the outside of the cell. The inside of the cell becomes more negative. This leads to the hyperpolarization of the membrane potential and therefore IPSP. Now, in summary, therefore, we can say that we have discussed about the presence of more than 50 neurotransmitters or their candidates, and some neurons are capable of releasing 2 or more neurotransmitters, and which are produced at different times depending upon the appropriate stimulation frequency. The synapses can be of two types. They can be electrical synapses, which allows direct exchange of ions between the pre and the postsynaptic cells via gap junctions, or chemical synapses, in which the neurotransmitters are released from the presynaptic cell, and these neurotransmitters sit on their respective receptor and initiate changes in the postsynaptic cell. The receptors on the postsynaptic cell onto which the neurotransmitter binds can be either channel-linked receptors, which lead to the opening of specific ion channels, or G protein-coupled receptors, which lead to the activation of a cascade of metabolic changes in the postsynaptic neuron. Now, binding to, depending on whether it is excitatory neurotransmitter, the binding to a excitatory neurotransmitter to the receptor on the postsynaptic membrane leads to the induction of EPSP in the postsynaptic neuron. On the other hand, the binding of an inhibitory neurotransmitter to its appropriate receptor leads to the induction of a IPSP or inhibitory postsynaptic potential in the postsynaptic neuron.